Hi, everyone. I would like to present you my recent work, which is called Somatem. So we're developing a long read focused metagenomics pipeline, as well as an accessible chat GPT kind of a co-pilot. And before I start, I would like to acknowledge all the other authors along with me on this paper. There's plenty of people, and also a lot of grants that have helped us get this early work started. Briefly, the outline of my talk, I lay down what the big problem it is we're trying to solve with this, and I'll go over the pipeline a little bit, and then talk a bit about how we are doing this co-pilot. So briefly, what we are calling the problem here is bioinformatics expertise gap. So the idea is that metagenomic data is generated a lot faster than researchers can analyze it. And why this happens is experimental biologists have a skill set that is very much geared towards determining the questions and then setting up experiments involving bacteria and microbial samples. And then they generate sequencing data through that. But once they have the data in hand, you still need to do some analysis to get the actual meaning out of the data. But a lot of experimental biologists, including me until a year ago, we typically lack the expertise in doing this bioinformatic analysis. So summarizing this in the image again, so you have your bacterial sample in hand, a microbiome sample, you put it through a nanopore sequencer, and you end up with this text file with data on it. You can stare at it as long as you want, but if you don't have the skills of using the command line and specialized software to analyze it, and especially since you have a big data set, can't analyze on your own computer. You need cloud computing skills or like connect to an academic computing cluster. So you need to do a lot of specialized things, and you need to process the data in a certain way. You need to clean up your reads, and you need to match it to certain databases and do statistics. So for you to get from the data to insight, there's a huge learning curve for a non-bioinformatician to learn from scratch. So what this causes is, okay, I'll give you the potential. So why are we worried about microbiome metagenomics kind of analysis? So let's say I'm a clinician and I have a patient in front of me, and they're presenting with an antibiotic resistant kind of bacteria. So I want to know what is infecting them and what antibiotics are they resistant to before I decide to treat them. So first thing, you collect the patient sample, then you analyze their sequencing with shotgun sequence that gives you the full genetic information of all the genes in the sample. Then you want to do a taxonomic profiling where you match it to a database of all the organisms. So you want to determine which species it is. So let's say there is Clostridium difficile, and you get to know how abundant it is in the population. But another thing you can know is also like, because of this infection, your overall microbial diversity is also reduced. So these are useful things if you know where to look. The next thing you would do is functional profiling. So you know the organism, you want to know within this organism's genes, is there any matches to a database of an antibiotic resistance gene or something? So you look for those genes. But for someone who is not from a computational background, a bioinformatic background, the bottleneck here is you want to articulate the things that you know in biological terms in the bioinformatic terms. So you need to know the bioinformatic name for the task. And then you need to know for that particular task, what are the tools that you can use? And there are many tools to choose from. So it's very overwhelming. Next thing is, once the tool runs and you get the output, you need to be able to interpret it. So you need to know some bioinformatics and some statistics for you to do that. So the kind of tools you would use is cleaning up the data. There's a bunch of tools, Nanoplot, Chopper. And taxonomic profiling, there's a whole suite of tools. And even functional profiling, there are many tools. So the choice is quite overwhelming. So our idea is, let's make a pipeline and then add like a LLM on it for people to reduce this kind of overwhelming choice and then ease the learning curve for getting into bioinformatics. So our solution here is, first thing, we want to develop a Nextflow pipeline, and we chose Nextflow because it was very modular and the framework was extensible. And we want to design a pipeline specifically for long reads, which is what we think the future of metagenomics is going to be. And we incorporate many publicly available NF Core modules. Thanks a lot for the NF Core community for these. Next thing we want to do is make a LLM copilot. So for a biologist who has like, they can articulate what they want to do in plain text. And the LLM takes care of translating this into the knowledge of the bioinformatic software. And then it also configures the software for you, and it has full knowledge of the pipeline. I'll come to what we mean by this in a minute. So we believe that a combination of creating our own pipeline and creating our copilot along with the pipeline is better than using a general purpose pipeline and general LLMs like ChatGPT, which are prone to hallucinations and are not like tailor-made for the pipeline that we have in mind. So briefly, the pipeline we have has like 20 plus modules, and in the Nextflow method, we chain the modules together into sub workflows and workflows. I'm showing a brief overview on the right here. So we have one sub workflow for initial QC, and then we have 4 choices. There are like 4 paths that we can go down into. They can either run all of them or only a few of them. So if someone is running on a laptop, they have like a light, we can only run the lightweight tasks on it. So taxonomic profiling, either with 16s or metagenomic reads. That's only one of the 4 branches here. But if you have like a workstation, like a academic cluster or something, you can run all of the tasks here, or you can choose the other tasks to run as well. So we include genome assembly, that includes metagenome mags, and pathogen or toxin genes detection, and also one thing where we analyze time series for structural variants in a recent software we have from our lab called RIA. So once you have the pipeline design, you would want to configure the pipeline. So we have a lot of default configurations that we provide. So if anyone just turns on the Nextflow and does not provide any extra things, it will run the same analysis for everyone. And that's not what people want to do ideally. So depending on the data, and hopefully with help from the LLM, they can configure this custom configuration in a different file format. They will choose which sub workflow they want to run, one or multiple of them. And they can also like change the parameters to make some analysis more stringent or something like that, depending on the environment that the sample is from, whether it's from soil, gut microbiome or something like that. So without any expert advice, the user would need to figure out what options to set here. And once we ship out, the pipeline is currently still private. Once we publish the paper in a couple months, it will be open source. So once someone has this on GitHub, what they will have to do is to clone it on their own computer. So initial setup, and they would have to download the Nextflow and all the dependencies with a Conda environment that we package and provide to people. And then they also have to choose all of their paths within the 4 databases in the Nextflow config. So this is where the databases will be downloaded by the Nextflow automatically. So once you do that, you need to figure out configuration details. So if you're looking through the default config, you need to be able to understand what the values in the config translate to what values in a particular software that you're using. So what do these mean in terms of the pipeline? And the next thing is, you need to be able to know what values in the software translate to what biology setting. So if my question is for a very high stringency setup, you need to know what threshold to increase and by how much to increase it. And if I'm okay with false positives, but I have a very low abundance sample, I want to be more generous in interpreting the data. You need to know what thresholds to change for that. So once you know all of these details, you would fill it out in a custom configuration file. And then you would feed it into Nextflow using a command where you say Nextflow, run this whole pipeline with this extra configuration that I just made. So for a new user, even just the first couple steps are quite overwhelming. But the part where they have to configure the pipeline by figuring out what their biological question is and what kind of data set warrants what configuration, that's like a huge overwhelming choice with a lot of Google search, reading many papers. So next we come to our Copilot, which is you would say one of the main innovations of this work, is we are hoping that this will enhance the usability of the Nextflow pipeline that we make. So the analogy that we like to come up with is, think about this Copilot chat interface as a way of talking to the pipeline developers. So if a developer is available on GitHub, you would go tell them, hey, I have like weird sample, it's not working, what do I do? So you can tell the same thing to the Copilot chat, and it will try to guide you through the process of like figuring it out by the extra knowledge that we code into it. So one thing it can help out is this is what we have like mostly implemented by now. So to configure and initialize and run the pipeline with the optimal parameters that match to the whatever is your main question that you're trying to identify. And also like a few key features of the data is like what kind of a microbial community it is coming from and how like rare the organisms are. So things like that. So it will help you pick a branch in the pipeline. And also if you have to run multiple branches and like mix the results or something, that is something it would tell you to do. So you can pick whether you need to assemble the genomes, assemble the reads into a genome, which is like a compute heavy step, or you can skip that and only do taxonomic profiling if your question only requires that. So you can also skip one extra step where you have to remove the host reads. If you have a gut microbial sample and you only care about the microbes, not the mouse that the microbes are from, you can remove those reads. But this will take a long time depending on how big your file is. So if you're not doing a gut microbiome sample, you can skip this. But if you didn't know that, you have to go read through the config and then, oh, figure out this is the step I need to skip. But the LLM can suggest you that, okay, this step needs to be skipped and it will automatically change it for you. So one thing is like picking out the things for you, but we don't want to treat this as a black box. So you want to be able to understand what analysis you're running. So it will also explain all the steps and choices that it has made for you. And you will get to finally approve that before it starts running the pipeline. And while doing all of this, it will also summarize. Hopefully we haven't done this part yet, but we are going to make it summarize all the choices made and the questions you've asked and the kind of like statistics that are involved in a short form that you can write on your own for your method section. So you can use this as a primer to write your method section, not copy pasted. And this is a future step, but one more thing that would be really cool is during the pipeline run. So some parts of the pipeline would have finished and then you would get your quality information while the assembly part is happening, which might take a few hours, like 4 to 5 hours. So while these intermediate results are already out, you can still sit and try to understand what they are doing. And if anything has been like something wrong has been done, you can stop working from there and start again by changing that parameter. So you don't need to waste the full time for the full workflow. So by not pausing, but like interrogating the results in the middle, if the LLM can help with this while the pipeline is running, this would be something really cool. And another thing is after the pipeline has run, if it can help us interpret the results and add it to the report that it's generating for the method section. And also if I do like multiple runs over a couple months, if I've ran like multiple samples, if I might be able to look at any outlier kind of samples within my whole batch, that would be like a meta-analysis that an LLM can probably help with. And this is also future task for us. So what this metagenomics copilot does is it translates your natural language, which would be like identify the species, compare healthy or diseased microbiomes, into executable pipelines, and it will execute the pipeline for you. Might take multiple hours, like 6-7 hours, but then you'll get a report which it will help you interpret as well. And all of this is powered by this thing called retrieval augmented generation. So I'll give you a brief intuition of what it is. So if you remember searching in a Google or some other search engine in the last maybe like 6 months or 1 year, you can recall that you get like a summarized output along with the citations. So the key part here is that it generates real citations that you can click and go through. So how it's doing this is you have your question, and if you type it into a normal Google, it will give you 100 web pages. So those 100 web pages have this information that the LLM is pulling out and summarizing for us. So basically that's what a RAG does. So if you replace the search engine with kind of custom knowledge that we put in, so we put in curated set of research papers. All the tools that we are using are from published open source papers. So we feed those into the knowledge base. And we also include like summaries of the GitHub README. So we have that. And then we also have a representation of the paths within our pipeline. So which tool is connected to which, and like what subset of tools does it make sense to run together. So we feed this information in. And then we also have another parameter where these are the definitions of what does each parameter mean. And this would be something useful for the LLM to know as well. It's like a distilled version of the knowledge that separates from what's in the research papers. So we feed them all of these, and we're hoping that using this copilot with these extra information would reduce hallucination, but it will also help people by reducing the learning curve. So it will democratize computational analysis. And we want people to learn by doing, so use this as a tutorial tool and not as a black box. And we also want to ensure people use best practices by making our own pipeline, which we will feed in with the best practices and the tools. So just to summarize, an experimental biologist has a few very valuable skills, but they lack the skill that translate the data into the insight. And this is where we come up with our own custom pipeline and collaborating with a domain-specific chatbot that we make. And we're hoping that this copilot, Somatem, will help people identify the tools, help with the installation, and also configuring the tools and running it. And in the future, we would want it to do more custom statistical analysis and visualization, but we're not there yet. But the copilot is useful because it has a custom knowledge base. It has access to command line tools. And with this, I'll stop my talk and take any questions. And there's a QR code link for a feedback survey if you want to leave any extended comments. Thank you.